Hi there, this is a brief presentation on Mendelian genetics. Everything that's in this presentation is essential to understand so that you can successfully perform Punnett squares. First of all, recall that genes are segments of DNA that code for particular traits, like whether or not you have freckles. Different versions of those genes are called alleles, so they're kind of considered like different options for a gene or a trait. Here are a list of some common human traits that we can see the different alleles for. We'll categorize them as either dominant or recessive. Dominant alleles are those that always show up if they're present, meaning that is the trait that is expressed. They will in fact mask the presence of a recessive alleles, and we denote them with a capital letter. So let's take, for example, freckles. There is a gene for the presence of freckles. If you have the dominant version of the gene, you have freckles. If you have the recessive version of the gene, you do not have freckles. Now, recessive, this is an allele that is hidden by the dominant allele, and we denote those by a lowercase letter. Another trait that's common in people is a widow's peak, where just in between your eyes, but at the top of your hairline, it comes to a point. So there's a kind of like a little point at the top of your hairline, whereas a straight hairline is recessive. Another fun one is whether or not you have a free earlobe or attached earlobe. And so if you can feel where the bottom of your earlobe attaches to your head, if it kind of makes a little U and you can kind of push it back and forth, that's a free earlobe, that's dominant. If it's attached and it goes straight into the side of your head, that's attached. So we denote recessive alleles with a lowercase letter. So think for a moment about what's the only way a recessive trait will show up. Here are a couple of other images for you to take a look at. The widow's peak here on the left is the dominant allele and the straight is on the right. And what you'll notice here are what we call genotypes, where we have two letters. One of each is representing an allele that you inherited from each parent. So the boy on the left, we would either say his genotype is two capital W's or capital W then lowercase w. Remember, you only need one copy of the dominant allele for that trait to show up. Whereas the boy on the right, who's pulling back his hair, has a straight hairline, which is the recessive condition. He must have two recessive alleles in order for that trait to show up. And so he has two little w's. Top right, you'll see dimples versus no dimples. No dimples is recessive. Having dimples is dominant. You can have it with one of two genotypes, two uppercase D's or uppercase D, lowercase d, and then again freckles, as we indicated on the last slide. Not having freckles is recessive, so you have to have two recessive copies, so one from each parent, in order to not have freckles. All right, moving on. Let's talk about genotype versus phenotype. There's a lot of important vocabulary in this lesson, and these are related, but they are different things. The genotype is the actual combination of alleles for a gene, like the W's, the F's, the D's that we saw on the last slide. Depending on what trait you're using, the letter may vary, but here you're seeing three different genotypes with the trait being denoted by a T. The phenotype is the physical appearance or result of the genes. So in this case, tall or short. Homozygous means that the two alleles that are in the genotype are the same. So they could both be dominant or they could both be recessive, but basically they are the same. So we see homozygous dominant with the two big T's and homozygous recessive with the little t's. We sometimes also call this a purebred. Heterozygous is where two different alleles exist in a genotype. So in this case, that's the big T, little t. And hetero means different, homo means same. Zygous refers to the zygote that is formed from the fertilization of an egg. Heterozygous individuals or for traits are also known as hybrids because they are a mix of the two. Here are some examples of genotypes and phenotypes, and the T's are referencing really what Mendel did in breeding of pea plants. In this particular short version of this lesson, we're not going through all of the details, but he was an Austrian monk who studied the genetics of pea plants and discovered that they carried very simple traits like tall versus short. They did not have in-between heights of plants. And so those individuals who were homozygous dominant were always tall. Those who were heterozygous, big T, little t, were also tall, but those who were homozygous recessive were short, little t, little t. Here are two laws that you should know. Each person has two alleles for each gene, as you might remember. One law is the law of segregation, which simply says that alleles will separate when making eggs or sperm, and so only one gets passed on from one cell into the gametes that are formed. So what percent of your genes would have come from your mom or your dad? Think about that for a moment. The other law is the law of independent assortment. This has to do with the idea that each gene is inherited independently of one another. So the inheritance of high eye color is not necessarily connected to the inheritance of skin color or hair color or eye color. Can't remember which one I first said. You should roughly get about 50% of your genes from each parent. So in other words, your hair color for law of independent assortment should not affect your eye color, assuming they are inherited independently. Let's take a look at a couple of crosses. So for Punnett squares, let's look at a homozygous times a homozygous cross. This is a model that we use to predict probabilities of a genetic cross and which traits will be inherited by each offspring. So the first thing you would do is write down the parental genotypes. And what you see on the top right is we're going to do a cross between a homozygous and homozygous individual. In this case, the P, that's parental, you're seeing a homozygous dominant and a homozygous recessive individual. Then we're going to find the possible gametes that they can generate. Remember, gametes are haploid. And so this is where we will use each of those genotypes and split them apart to figure out the types of gametes they can make. And then we'll use a Punnett square to find the possible offspring genotypes, which would be back to diploid. So given our parental crosses of big P, little p, those are diploid, we need to figure out the haploid gametes each can make. So in this case, our big P purple flower, so the two big P's, this is our flower in the bottom right here, the purple one with the two big P's. When this diploid genotype splits into haploid genotypes, there's only one kind of gamete that this flower can make. They will all be little, big P's. The white flower, little p, can only make one kind of gamete as well. These would be lowercase p's. So we separate these into potential gametes that they can make. Then we recombine them from each parent in each box. So this top left box is a combination of the big P from the purple flower and a little p from the white flower. Scooting over to the top right box, we recombine the right hand little p and the top big P to form big P, little p. And you continue on the bottom left and the bottom right. In this case, because both parents were purebreds, we can only make one kind of offspring, big P, little p. So they are all purple, but notice they're all heterozygous, so they have different genotypes than both parents. So we could say that our genotypic ratio would be 4 big P, little p to 0 anything else. The phenotypic ratio would be 4 purple to 0 white. Let's look at two more crosses. We'll do a homozygous and heterozygous one with, let's consider the fact that black will be dominant to brown. And here is our cross. Here we have some guinea pigs. It's kind of tough to tell that these guys are black. It's an old image. But you have a black guinea pig who is homozygous dominant crossed with a black guinea pig who is heterozygous. Still black, but heterozygous. When we cross these individuals, we separate the genotypes into gametes. So the top guinea pig, half of the gametes will be big B's, half of their gametes will be little b's. Remember, when we say gametes, we mean sperm or egg cells. Then we take the homozygous black guinea pig and separate these into its gametes. Then we recombine them into our Punnett square. Top left, big B, little b, one from each parent. Top right, big B, little b. And then we see the same repeating pattern on the bottom. So when we break down our genotypic ratio, notice how we add in the genotypes. We have 2 big B's, This is the F1 is the first filial generation, meaning the offspring. And then we end up with 2 big B little b's, or 2 heterozygotes. Now because this ratio it's 2 to 2, you could divide that and get a 1 to 1 ratio, and that's still the same ratio. Then when we look at our phenotypic ratio, we have all black guinea pigs because all of these have a dominant allele. So 4 black to 0 brown. Now let's look at one last cross, which is a little more exciting, heterozygous to heterozygous. Here we're going to take 2 brown rabbits. They're both excuse me, let me repeat myself. These are 2 black rabbits who are both heterozygous. Again, black dominant to brown. They're both heterozygous. So even though they are black, they carry the brown allele, much like this guinea pig up here did, except that it reproduced with a homozygous dominant other parent. So there's no brown guinea pigs. Here we have 2 black rabbits that each carry the brown allele. So when we cross, this is our parental cross of big B little b times big B little b. When we separate these into gametes, this rabbit down here, let's say it's the male, half of the sperm would be big B's, the other half would be little b's. And if this was the female, half of her eggs would carry big B, the other half would carry little b. So when we recombine these gametes, so egg to sperm, this proportion of the offspring, 1/4 or 25%, would be homozygous dominant. Then we see 2 boxes out of the 4 that would be heterozygous. Big B, little b. Big B, little b. So half of the boxes, or 50%, would be heterozygous, still black. Then 1/4 of the boxes, notice here now we do get brown rabbit showing up, little b, little b. So notice here we have some interesting ratios. Genotypic ratio, we have 1 homozygous dominant, big B, to 2 heterozygotes, big B, little b, to 1 homozygous recessive, little b. But the phenotypic ratio is a little more interesting, where we have 3 black to 1 brown. And so you have to interpret the 2 ratios separately from one another because they're not always the same. Hopefully this is helpful and enough to get you going on your practice.